Okay, hello everyone. This is my first attempt at a show me, so be kind please. So we're looking at the structure of DNA today. Now, DNA is a macromolecule, meaning it's very, very big, and DNA also carries our genetic code. That meaning it's divided, each strand of DNA divided into segments called genes, and each gene codes for a particular protein. But you should know this already from Nat5. The DNA molecule is made of two complementary strands, and we'll have a look at those in a bit more detail later. And those strands are joined together and create this pattern that you can see in the picture here called a double helix. Not a spiral, double helix. So each of these two strands that make up the DNA molecule, the actual lengthwise parts of those strands are the sugar phosphate backbones. So on this diagram here, the sugar phosphate backbone for one strand would go like this. And there's the other complementary strand opposite. Okay, so the strands twist to create the double helix pattern. And linking the sugar phosphate backbones together, we have base pairs, and they would form these parts of the strand going across between the sugar phosphate backbones, like so. So that's where the bases would be in the DNA molecule. Okay, so now if we look at the basic component of the DNA molecule, that is called a nucleotide, and the structure of a nucleotide looks like this. Now the blue molecule, the pentagon-shaped molecule in the middle, is deoxyribose sugar. Now the green molecule up in the top left is a phosphate group. And the red part of the molecule here is a base. You can call it a nitrogenous base, it can be called an organic base, but just the word base will do. Now those bases can be either adenine, thymine, guanine, or cytosine. And we'll have another look at those bases a bit later on. Now, something very important that you need to know in terms of the structure of the nucleotide is the 3' and the 5' ends of that nucleotide, and that's very important when they link together. Now, if we imagine on this ribose sugar, ribose is a 5-carbon sugar, so let's highlight those 5 carbons. So there is the 1, and there's number 2, there's number 3, there's number 4, and there's number 5. So we call this carbon here the 3' carbon, and this one up here the 5' carbon. So the 3' is at the bottom of the picture as you see it now, and the 5' is at the top. Okay, so let's have a look at the DNA structure with regards to the nucleotides in a bit more detail. But first of all, we'll build up two chains of complementary nucleotide strands. So we have our two complementary strands, and now we're not looking at it as though it's a double helix anymore. We're looking at it as though it's the helix has been spread out flat, a bit like a ladder. So what we have here are our sugars, deoxyribose sugars. That's those there. And here's our phosphate groups with the green dots inside them like so. And here are the bases forming in our picture what would be the rungs of the ladder, as it were. Okay, so now what you see about these two complementary DNA strands, strands of nucleotides, is that one of the strands seems to run in that direction from the 3' end to the 5' end, and the other strand seems to be upside down, running in the opposite direction. So the 3' end would be up there and the 5' end down there. Now, because of that arrangement in the DNA molecule, we say that the two complementary strands are anti-parallel strands, and also that in between the sugars on the phosphates and the sugar phosphate backbones of each strand, there are strong chemical bonds that hold them together, and in between the bases there are basic hydrogen bonds that will hold the bases together. Okay, so they would be there in between the bases of the two strands. Okay, so finally, if we have a look at how those bases pair up together, it is like so. Adenine will pair up with thymine, and guanine will pair up with cytosine. This is the base pairing rule, and of course can be expressed either using the full names or as ATGC, like so. Now, the reason why that is the case, and you don't really need to know this, it's just a bit of interesting information to understand why they pair up the way they do, is that if you look at the hydrogen bonds between the molecules, adenine and thymine form 2 hydrogen bonds, but guanine and cytosine will form 3 hydrogen bonds. So if cytosine were, for example, to try and bond with adenine, because of that mismatching in terms of the ability to make hydrogen bonds, they wouldn't be able to come together and align themselves properly to form the correct structure of the DNA molecule. So it's the formation of those hydrogen bonds and the ability to form those different numbers of hydrogen bonds guarantees that adenine and thymine and guanine and cytosine pair up with each other. Okay, so that is the basic introduction to the structure of DNA. Thank you very much.